Students, this is Shahada here, Department of Zoology, Maharani Science College for Women, Bangalore. So in this session, we are going to discuss about the cell cycle and regulation under Unit 4 Cell Biology. Moving on to the contents here, we shall discuss about an introduction to cell and the definition of cell cycle, stages of cell cycle, regulation of cell cycle. Before going to explain what is cell cycle and regulation, let us understand what is the meaning of cell. The cell is the basic structural, functional and biological unit of all the living organisms. Cells are the smallest units of life, hence are often referred to as the building blocks of life. So here I have the picture for the cell here having different parts like nucleus, ribosome, rough endoplasmic reticulum, plasma membrane, cell coat, cytoplasm, Golgi body, mitochondria, centriole, free ribosome, smooth endoplasmic reticulum, lysosome, nuclear pore, chromatin, nucleolus and nuclear envelope. So these are the different parts which make up the cell. Again, you have already known the function of each part of the cell. A cell usually consists of a nucleus and the cytoplasm and is contained within the cell membrane, which contains many biomolecules such as proteins and nucleic acids. To divide, a cell must complete several important tasks. It must grow, copy its genetic material, and physically split into two daughter cells. Cells perform these tasks in an organized, predictable series of steps that make up the cell cycle. The cell cycle is a cycle rather than a linear pathway because at the end of each go round, that is, the two daughter cells can start the exact same process over again from the beginning. So the most basic function of the cell cycle is to duplicate accurately the vast amount of DNA in the chromosomes and then segregate the copies precisely into two genetically identical daughter cells. Now let us see what is the exact definition of cell cycle. The cell cycle or cell division cycle is the series of events that take place in a cell that cause it to divide into two daughter cells. These events include the duplication of its DNA, nothing but DNA replication, and some of its organelles and subsequently the partitioning of its cytoplasm and other components into two daughter cells in a process called cell division. The cell division cycle is a vital process by which a single-celled fertilized egg develops into a mature organism, as well as the process by which hair, skin, blood cells and some internal organs are renewed. So this is the definition of cell cycle. Now moving on to the stages of cell cycle. In cells with the nuclei, often called as eukaryotes, that is, animal, plant, fungal and protist cells, the cell cycle is usually divided into two main stages, that is interphase, which is again divided into three phases, that is G1 phase, S phase and G2 phase. This interphase is continued with the mitotic phase or M phase, which is divisible into two phases, that is karyokinesis and cytokinesis. Now let us see in detail. The first stage in the cell cycle is the interphase, which is a series of changes that takes place in a newly formed cell and its nucleus before it becomes capable of division. Again, it is also called as preparatory phase or intermitosis. So during interphase, the cell grows, accumulates nutrients needed for mitosis and replicates its DNA and some of its organelles. Next, moving on to the stages of interphase, which proceeds in 3 stages: G1, S and G2 phases. So before entering into G1 phase, the cell is at resting phase called as G0 phase. So G0 phase is a resting phase where the cell has left the cell cycle and has stopped dividing. The cell cycle starts with this phase. Some cells which do not divide further attain an inactive G0 phase, also known as quiescent phase, after they exit G1 phase. G0 phase is a permanent state for some cells, while others may restart division if they get the right signals. So this is the G0 phase, which is the starting point of interphase, which is also known as the resting phase. So here I have the picture for the cell cycle where you can clearly observe the different events in the cell cycle. So the cell cycle usually starts with the G0 phase, which is known as the resting phase. So when it is triggered by the triggers, like it enters into the G1 phase and then to S phase and enters into G2 phase, and finally it comes to the mitotic phase or the M phase, which is again divisible into the karyokinesis and the cytokinesis, and the resulting products are 2 daughter cells. So this is nothing but the cell cycle. Next, moving on to the stages of interphase. The first one is the G1 phase or growth phase. It is also called as the first gap phase. It is the first phase within the interphase from the end of the previous M phase until the beginning of the DNA synthesis is called G1 phase. In this phase, the cell grows physically larger in preparation for DNA replication, and cellular contents are also duplicated in this phase. The duration of the G1 phase is highly variable even among different cells of the same species. So this is nothing but the G1 phase of the interphase. So where the cell physically gets larger and prepares for DNA replication. So here I have the picture for the G1 phase, having the labeling such as the centrosome, the nuclear envelope, nucleolus, chromatin and cytosol. So here the DNA will be in the form of chromatin. So there is no structures like chromosome you can find in this stage. So the DNA material will be in the form of chromatin. So next, moving on to the S phase. In this phase, the cell synthesizes a complete copy of the DNA in its nucleus. It also duplicates a microtubule organizing structure called the centrosome. So the centrosomes helps to separate DNA during M phase. So throughout the interphase, the nuclear DNA is in the form of semi-condensed chromatin. But in the S phase, the DNA replication can proceed through the mechanism that results in the formation of identical pairs of DNA molecules called as the sister chromatids. So in the picture you can clearly observe the chromosomes having the sister chromatids that are firmly attached to the centromeric region. The centrosome is duplicated during the S phase, and these centrosomes will give rise to the mitotic spindle, the apparatus that orchestrates the movement of chromosome during mitosis. So this is the S phase where you can have the DNA synthesis. So the G1 phase is the growth phase and the S phase is the synthesis phase of the DNA, where you can see the sister chromatids having the centromere, which leads to the production of spindle fibers. Next is the G2 phase. The G2 phase occurs after DNA replication and is a period of protein synthesis and rapid cell growth to prepare the cell for mitosis. During this phase, microtubules begin to recognize to form spindle fibers. Before proceeding into mitotic phase, cells must check at the G2 checkpoint for any DNA damage. Within the chromosome, the G2 checkpoint is mainly regulated by the tumor protein, that is P53. If the DNA is damaged, P53 will either repair the DNA or trigger the apoptosis of the cell. If P53 is dysfunctional or mutated, cells with damaged DNA may continue through the cell cycle, leading to the development of cancer. So this is nothing but the G2 phase. So in this phase, the cells grow rapidly and they prepare for the mitosis. So in this picture you can see the spindle fibers exactly like the picture having the labeling such as the chromosome, the centrosome here, and the spindle fibers, which again is the indication of mitosis. Next, the G1S, G2 phases together are known as interphase. So we have already discussed this. The prefix inter means between, reflecting that interphase takes place between one mitotic phase and the next. Next, moving on to the mitotic phase, which is known as M phase or chromosome separation. The M phase or mitotic phase of the cell cycle is the most crucial and dramatic phase of the entire cycle, where the cell divides to form two identical daughter cells. The most important event of this phase is the karyokinesis, that is nothing but the nuclear division, where the chromosome separates into form two distinct cells. During the mitotic phase, the cells divides its copied DNA and cytoplasm to make two new cells. So we have already discussed M phase involves two distinct division related processes, that is karyokinesis, which is known as nuclear division, and the second one is cytokinesis, that is nothing but division of cytoplasm. Now let us see what is karyokinesis. Karyokinesis is also known as mitosis, consists of a series of phases, that is prophase, prometaphase, metaphase, anaphase and telophase, that results in the division of the cell nucleus. It is the first step during the mitosis and result in the formation of two daughter nuclei, which corresponds to separation of daughter chromosomes into these two nuclei. It is generally followed by cytokinesis. So here I have the picture for the karyokinesis, that is the first mother nucleus is dividing into two daughter nuclei. So that is nothing but the division of the nucleus into two daughter nuclei is called as karyokinesis. Next is cytokinesis. Karyokinesis is immediately followed by cytokinesis, which divides the nuclei, cytoplasm, organelles and cell membrane into two cells containing roughly equal shares of these cellular components. Mitosis and cytokinesis together define the division of the mother cell into two daughter cells, genetically identical to each other and to their parent cell. So this is nothing but the cytokinesis, where you can have the complete division of the two daughter cells having the equal amount of the DNA material. So in this picture you can clearly observe the two daughter cells having the complete set of DNA material. Okay, so this is the furrow where upon division it will have two daughter cells. So this is again the picture of the cell cycle, where you have the G1 phase, gap 1 phase, and S phase, that is the synthesis phase, and G2 phase, where the cells with the duplicated chromosomes, and after that it enters into the mitosis, and that is nothing but the chromosome separation, and the next stage is cytokinesis, where you have the division of cells, that is the two daughter cells. So that is nothing but the cell cycle. Next, moving on to the regulation of the cell cycle. The cell cycle does not occur in unchecked manner and needs to be highly controlled, as uncontrolled division might give rise to diseased states such as cancer. The preparation of the cell cycle are checked by regulatory molecules. These regulatory molecules detect and repair the DNA, that is the damaged DNA, as well as prevent the uncontrolled cell division. These regulatory molecules may act individually, or they can influence the activity or production of the other regulatory proteins. Therefore, the failure of a single regulator may have almost no effect on the cell cycle, especially if more than one mechanism controls the same event. Conversely, the effect of a deficient or non-functioning regulator can be wide-ranging and possibly fatal to the cell if multiple processes are affected. Regulation of the cell cycle is divisible into two types. First is cell cycle checkpoints, and the second is regulatory molecules. Now let us see what are these cell cycle checkpoints. These are the internally controlled checkpoints, where the cell examines internal and external cues and decides whether to move forward with the division or to stop. So these checkpoints clearly notices if any internal and external cues where it can be proceeded forward or to stop, like that. So there are number of checkpoints, but the three most important ones are the G1 checkpoint, the G2 checkpoint, and the spindle checkpoint. So the G1 checkpoint is at the G1 and S transition. The G2 checkpoint is at the G2 or M transition, and the spindle checkpoint at the transition from the metaphase to anaphase. So here I have the pictures for the G1 checkpoint, which checks for the cell size, nutrients, growth factors and DNA damage. And the G2 checkpoints which check for the DNA damage, DNA replication and etc. And also the spindle checkpoint which checks for the chromosome attachment to the spindle at metaphase plate. So this is nothing but the spindle checkpoint. Next is the regulatory molecules. So in addition to the internally controlled checkpoints, there are two groups of intracellular molecules that regulate the cell cycle. These regulatory molecules either promote progress of the cell cycle to the next phase, that is nothing but the positive regulation, or halt the cell cycle, that is nothing but the negative regulation. Now let us see what is positive regulation. So in positive regulation, active molecules cause the cell cycle to progress. So there are two key classes of positive regulatory molecules that determine a cell proper progress through the cell cycle through various checkpoints. So for the first is the cyclins and the second is cyclin dependent kinases, which is known as CDKs. So let us see what is cyclin. Cyclin is a family of protein that controls the progression of cell cycle. These cyclins regulate the cell cycle only when they are tightly bound to the CDKs, that is the cyclin dependent kinase. So to be fully active, these CDK and cyclin complexes must also be phosphorylated in specific location. So there are four types of cyclins. First is D-cyclin at G1 phase, cyclins E and A at S phase, and B-cyclin at M phase. So the concentration of these cyclins usually remains low for the most part of the cell cycle, but peaks dramatically if they are needed during the cycle. Next, cyclin dependent kinases are a group of enzymes that work to regulate different processes in the cell cycle. Cyclin dependent kinases are protein kinases that, when fully activated, can phosphorylate and activate other proteins that advance the cell cycle past a checkpoint. So there are usually three types of cyclin dependent kinases. The first one is CDK4 at G1 phase, CDK2 at S phase, CDK1 at M phase. Now that is about all the positive regulation where the active molecules progresses the cell cycle. Next, moving on to the negative regulation, that is the second group of cell cycle regulatory molecules are negative regulators. These negative regulators halt or stop the cell cycle from progression. Some of the well-known negative regulator molecules are retinoblastoma protein, also known as RBP, and second one is P53 and third one is P21. These negative regulatory molecules act primarily at the G1 checkpoint and prevent the cell from moving forward to division until damaged DNA is removed. Now let us see one by one what is retinoblastoma protein. Retinoblastoma proteins are negative regulators of the G1 to S cell cycle transition. It is a tumor suppressor gene which plays pivotal role in negative control of the cell cycle and in the tumor progression. So there are three types of RBP. The first one is P105, second is P107, third one is P130. So these are the different types of retinoblastoma protein which can stop the cell cycle. Next is P53, which is known as tumor protein P53, also known as P53 or tumor suppressor P53. So P53 halts the cell cycle and recruits enzyme to repair damaged DNA. If DNA cannot be repaired, P53 triggers apoptosis to prevent duplication. Mutations in the P53 gene may cause cancer cells to grow and spread in the body. So this is nothing but the gene P53 which can stop the cell cycle if there are any DNA damage occur in the cell cycle. Next is P21. The production of P21 is triggered by P53. The P21 halts the cell cycle by binding to and inhibiting the activity of the CDK and cyclin complexes. P21 is a cell cycle inhibitor that promotes both CDK and cyclin inhibition as well as cell cycle arrest during the G1 or S phase. In human, P21 is encoded by the CDKN1A gene on chromosome 6. So this is about today's session where we discussed about the cell cycle and the stages of cell cycle and the regulation, where we discussed about the cell cycle checkpoints and the regulatory molecules. So in the regulatory molecules we discussed about two types, the positive regulators and the negative regulators. The positive regulators are the regulators which progresses the cell cycle, where we discussed about two cyclins and the cyclin dependent kinases. And the negative regulators we discussed about the RBP protein P53 and P21 gene. So these are the different types of regulators which regulate the cell cycle in the organisms. So thank you, students.